In this video tutorial, I am going to discuss about beta oxidation of fatty acid. Let's first look at the structure of fatty acid. So, structure of fatty acid is simply a hydrocarbon chain. So, CH3, CH2, CH2, so many CH2 gets repeated, once again CH2, CH2, and this hydrocarbon chain has a carboxylic acid group at one end. Now, depending on the number of carbons, fatty acid can be divided into four groups. It can be small chain or short chain, or it can be medium chain, it can be long chain, or it can be a very long chain fatty acid. Now, as there are many carbon, we need a proper system to refer to the carbon. So, there is a carbon numbering pattern. There are three different types of carbon numbering pattern. If we start counting carbons from this carboxyl group, we can count it as a delta 1, delta 2, this carbon is a delta 3 carbon, and numbers increases on this side. If we count carbon from this methyl end, it's called as a omega numbering system. So, this carbon is omega 1, this carbon is omega 2, this carbon is omega 3, and numbers increases on this side. The third system is little bit unique. It does not utilizes number, instead it utilizes Greek alphabet. In the Greek alphabet system, this carboxyl carbon is not given any identification. This carbon next to the carboxyl group is given a alpha carbon. This next carbon becomes a beta carbon. The next carbon becomes a gamma carbon, and alphabets increases on this side. So, these three numbering systems are there. Now, what happens? In our body, fatty acids can be oxidized many different way, but the major type of oxidation which occurs in our body is on this carbon, that is delta 3 or beta carbon. So, that carbon's oxidation is a major importance in our body, and it is accordingly called as a beta oxidation. And in this video, we will be describing beta oxidation of fatty acid. One more thing, that whenever required, I will draw a fatty acid structure, but I will not draw this entire fatty acid structure. Rather, what I will do, I will explicitly show these last three carbons, and remaining carbon skeleton of this fatty acid will be shown as a R group. So, this R represent this many carbons, that is the aliphatic chain, followed by this three carbon. So, we have CH2, CH2 followed by carboxyl group. So, this shorthand representation will be utilized whenever required for fatty acid. Now, let's start our discussion about fatty acid oxidation. First and most important question is, what is the location of beta oxidation of fatty acid? Remember, beta oxidation of fatty acid occur in almost all the tissues. However, there are certain exceptions in which beta oxidation does not occur, and these tissue are brain, red blood cell, or you can call it as RBCs, and adrenal medulla. At these three locations, beta oxidation does not occur. Now, inside the tissue, there are cells, and inside the cells, where does this beta oxidation of fatty acid occur? So, for that, suppose this square represents a cell. In the cell, there are many organelles. So, let's draw one organelle, that is mitochondria. Suppose this is the mitochondria. Mitochondria has two membrane. One is outer mitochondrial membrane. So, this is the outer mitochondrial membrane, shortly called as a OMM, and this is the inner mitochondrial membrane. So, this becomes our inner mitochondrial membrane. The space between these two membrane, that is between outer mitochondrial membrane and inner mitochondrial membrane, this space is called as a intermembrane space, whereas the space inside this mitochondria, it is called as a mitochondrial matrix. Now, what happens? Fatty acids are the cytosolic compound. This vacant space represents cytosol. Okay, so fatty acids are present in the cytosol, whereas all the enzymes which is required for the beta oxidation of fatty acid, they are there inside this mitochondrial matrix. So, enzymes of beta oxidation are in the mitochondrial matrix. So, what happens? Fatty acid is not permeable through this membrane. So, fatty acid simply cannot enter inside the mitochondria. So, for that, fatty acid has to first undergo activation, and fatty acid must be converted to the active fatty acid. Active fatty acid is also called as acyl coenzyme A. Once it is activated, then only it can enter inside this mitochondria by crossing outer mitochondrial membrane and inner mitochondrial membrane. And once it is inside this mitochondrial matrix, at that time only beta oxidation can be carried out. So as you can see, beta oxidation of fatty acid is actually a continuous process, but purely for the description purpose, we can divide this entire process into the three stages. The first stage is the activation of fatty acid. So let's write it down, that is activation of fatty acid. In the second stage, we describe that how this activated fatty acid enters or transported inside this mitochondrial matrix. So stage 2 is the transport of fatty acyl coenzyme A. Remember, it is also called as a fatty acyl coenzyme A, or you can simply call it as a acyl coenzyme A. So transport of acyl coenzyme A to mitochondria. Okay, and in the stage 3, once acyl coenzyme A is inside the mitochondrial matrix, we describe all the stages of beta oxidation of fatty acid. So in the stage 3, we describe beta oxidation proper. So let's start our discussion with the stage 1, that is activation of fatty acid. Now to describe this stage, I have to zoom in on this part. So here what happens? This is the cytosolic portion, this is the outer mitochondrial membrane, this is the intermembrane space, this is the inner mitochondrial membrane, and here is the mitochondrial matrix. So for that, suppose this is the cytosol. Right. Now, after the cytosol, I have to show outer mitochondrial membrane. So, suppose this band, let's color it. This band represents outer mitochondrial membrane. After outer mitochondrial membrane, we have intermembrane space. After intermembrane space, we have inner mitochondrial membrane. So, let's say this band represents inner mitochondrial membrane, and let's color it down. So this is the inner mitochondrial membrane, and below this inner mitochondrial membrane, I describe mitochondrial matrix where beta oxidation proper occurs. So currently in the cytosol, what we have? We have fatty acid. So the structure of fatty acid in the shorthand is going to be R, then 2CH2 group, and then followed by carboxyl group. So COO minus. This is the structure of fatty acid. Now in the stage 1, we have to activate the fatty acid. Now fatty acid activation involves a participation of coenzyme A. So this is a coenzyme A, right? Now in the coenzyme A, what happens? There is one very important SH group, and that is so important that we have to represent SH group into this shorthand notation of coenzyme A. So generally it is written as a coenzyme A SH. Remember, SH is not another part, rather it is integral part of coenzyme A. So this coenzyme A reacts with this fatty acid. Now this coenzyme A has special attraction towards this carboxylic carbon, right? So what happens? This coenzyme A attaches with this particular carbon through its sulfur. So let's write down the resultant product. So we have R, then we have 2CH2 group. Instead of COO, I will write C double bond O. One oxygen is incorporated but not shown in this diagram, and through the sulfur coenzyme A bounds. So sulfur followed by coenzyme A, and this resultant compound is called as a acyl coenzyme A. Now this reaction requires energy. That energy is provided by ATP. So ATP is broken down. It's two high energy bonds are used. So ATP gets converted to adenosine monophosphate plus inorganic pyrophosphate. As soon as inorganic pyrophosphate is generated, it is immediately broken down to 2 inorganic phosphate, and this reaction is catalyzed by the enzyme inorganic pyrophosphatase. This immediate breaking down of inorganic pyrophosphate ensures that this reaction remains irreversible. We also know that whenever in any reaction ATP are being participated, it requires magnesium ions. Now this particular reaction, as I told you the reason, it is an irreversible reaction, and this reaction is catalyzed by the enzyme which is actually present on this outer mitochondrial membrane, and the name of enzyme is acyl coenzyme A synthetase. The enzyme is acyl coenzyme A synthetase, or its other name is thiokinase. Now, this is surprising, right? Because this reaction takes place in the cytosol, but its catalytic enzyme is located inside this outer mitochondrial membrane. So, let's say this represents the enzyme. It is protruding at both the side, on the outer leaflet as well as on the inner leaflet of outer mitochondrial membrane. So, this enzyme catalyzes this first reaction. There is two peculiar thing about this enzyme. One thing is that this enzyme is not only present in outer mitochondrial membrane, rather it is also present in endoplasmic reticulum as well as peroxisomes. And second important thing is that there are in our body actually three different types of this enzyme is present. One type is specific for short chain fatty acid, another enzyme is specific for medium chain fatty acid, and third type is specific for long chain and very long chain fatty acids. And now this acyl coenzyme A is formed. It is also called as a active fatty acid, right? So with the formation of this active fatty acid, our stage 1 gets completed. Now let's look at the stage 2. What is stage 2? That is the transport of fatty acyl coenzyme A or acyl coenzyme A to the mitochondria. So stage 2, that is transport of acyl coenzyme A to mitochondria. Now here what happens? This acyl coenzyme A is freely permeable across the outer mitochondrial membrane. So acyl coenzyme A comes inside this intermembranous space. Now from this acyl coenzyme A needs to get transported across this inner mitochondrial membrane. But for short chain and medium chain fatty acid derived acyl coenzyme A is actually freely permeable across this inner mitochondrial membrane. But this is not the case with the long chain fatty acid derived acyl coenzyme A. Long chain derived acyl coenzyme A cannot get transported across this inner mitochondrial membrane. The problem is this coenzyme A. It does not let this acyl coenzyme A to pass through this inner mitochondrial membrane. So what happens? Acyl coenzyme A reacts with one separate molecule, that is carnitine. So it reacts with the carnitine, and what happens? Carnitine replaces coenzyme A. So what happens? This coenzyme gets released. So we write coenzyme A with its SH group, and in place of coenzyme A, carnitine gets attached. So resultant compound is the acyl carnitine. Now this reaction is catalyzed by the enzyme, and again this enzyme is also present in the outer mitochondrial membrane, and the name of this enzyme is the carnitine acyl coenzyme A transferase 1. So carnitine acyl CoA transferase 1, and this is also a outer mitochondrial membrane protein. It is protruding on both the side on the outer leaflet as well as inner leaflet of outer mitochondrial membrane, and this particular enzyme catalyzes this reaction. Once it is converted to acyl carnitine, then in the inner mitochondrial membrane there is a specific transportation channel. So suppose this is that particular transportation channel, and this transportation channel is actually of a counter mechanism, that acyl carnitine enters into the mitochondrial matrix. Acyl carnitine enters inside this mitochondrial matrix in exchange of carnitine. So carnitine travels from the mitochondrial matrix and it comes into the intermembrane space, and this carnitine can be reutilized over here. Now what happens? Now before that, one thing, that this transportation channel is actually an enzyme, okay, and the name of this transportation channel is the translocase. If you remember, generally for enzyme we describe that enzymes are divided into 6 different classes, but recently 7th class of enzyme has been described, and that 7th class is of the translocase variety. This translocase, what they do, they transport molecules across the membrane. So this is such class 7 of enzyme commission, and the complete name of translocase is based on what they transport. So on one side they transport carnitine, on another side they transport acyl carnitine. So this translocase is actually a carnitine, acyl carnitine translocase. Okay, so with the help of this translocase enzyme, acyl carnitine enters inside the mitochondrial matrix. But what we need in the mitochondrial matrix? We don't need acyl carnitine, we need acyl coenzyme A. So now again what happens? Here this acyl carnitine undergoes a reaction which is exactly reversal of this reaction which occurs in the intermembrane space. So here what happens? Coenzyme A with its SH group gets added into this reaction, and so now coenzyme A replaces the carnitine. So carnitine gets released and it gets transported across this membrane to appear in the intermembrane space. So now acyl bound with, instead of carnitine, it binds with the coenzyme A. So we get acyl coenzyme A. Remember, this entire route is required for the long chain fatty acid derived acyl coenzyme A. For the short chain and medium chain, they can be directly transported across this inner mitochondrial membrane. So all long chain, medium chain and short chain, all the, and of course very long chain also gets entered into the mitochondrial matrix. So with this it completes our description about the second stage, that is the transport of acyl coenzyme A to the mitochondria. Now once we have acyl coenzyme A inside this mitochondrial matrix, we can describe the stage 3, that is the beta oxidation proper. So the stage 3, that is beta oxidation proper. But before that, here I forgot to mention one thing, that this reversal reaction is catalyzed by the enzyme carnitine acyl coenzyme A transferase 2. Now this enzyme is located inside this inner mitochondrial membrane. Unlike this previous two enzyme, this enzyme is present in the inner mitochondrial membrane, and second thing, that it is not protruding on both the side, rather it is protruding only in the inside. So the name of this enzyme is carnitine acyl coenzyme A transferase 2. So this particular enzyme catalyzes this reaction. And here one thing very important, that this carnitine, what is the role of carnitine? It is actually acting like a car. This car takes up a passenger of acyl coenzyme A and get it transported across this border of inner mitochondrial membrane, and once it that passenger reaches to its destination, this carnitine returns back to its original starting position. So now it's time to discuss about the beta oxidation proper. So for beta oxidation proper, we have our acyl coenzyme A inside the mitochondrial matrix. So let's first draw a structure of acyl coenzyme A. So it is R group followed by 2CH2 followed by carboxylic carbon and then sulfur with its coenzyme A attached. This is the acyl coenzyme A. And remember, this is the alpha carbon and this is the beta carbon, right? Now we are describing what? We are describing beta oxidation. So this is the carbon, main carbon target for the oxidation. In the first reaction itself, oxidation takes place. Now in this oxidation step, what happens? One hydrogen from beta carbon and one hydrogen from the alpha carbon is lost, and it is lost to the FAD coenzyme. So FAD comes and takes away two hydrogen. So FAD gets converted to FADH2. This FADH2 further undergoes electron transport chain and can generate ATP. Now, how many ATPs are generated from FADH2? As per new information or new literature, 1.5 ATPs are generated. But if you are reading old book, as per old literature, it can generate 2 ATP. Okay. Now, once these two hydrogens are removed, what happens to the structure of this acyl coenzyme A? Let's draw it. So we have R group which is unchanged. On the CH2, one hydrogen is lost, so I will write CH. On this alpha carbon also one hydrogen is lost, so I will write CH over here. Now as both hydrogens are lost, this bond will become double bond to make carbon's valency for 4. Then remaining structure remains unchanged. So C double bond O followed by S and then coenzyme A. This reaction is catalyzed by the enzyme. In the name of enzyme, we generally write the name of substrate followed by its action. So here the action is dehydrogenase. So the name of enzyme is going to be first name of substrate, that is acyl coenzyme A followed by its action, that is dehydrogenase. Now this acyl coenzyme A dehydrogenase, there are actually 4 different variety in our body. What are these 4 different variety? One is specific for short chain, another is specific for medium chain, the next is specific for the long chain, and fourth one is specific for very long chain fatty acid derived acyl coenzyme A. Now this product, what should be its name? Remember earlier we are calling it as a acyl coenzyme A. But whenever there is a double bond in the backbone of this fatty acid, instead of acyl, we generally use word enoyl. So this is a enoyl which is bound with the coenzyme A. So enoyl coenzyme A. Now whenever there is a double bond in the structure of fatty acid, it needs to be specified 2 things. One thing is about its configuration, whether it is cis or trans. Here it is a trans configuration. So we write trans enoyl coenzyme A. And the second thing that needs to be specified is the location of double bond. And for the location of double bond, we use delta carbon numbering system. So this is a delta 1 carbon, this is delta 2, and this is delta 3 carbon. So the double bond is between delta 2 and delta 3. Here the small numbers is utilized. So this is a delta 2 trans enoyl coenzyme A. I think by this, if you remember the name, it will not be difficult. Now what happens to this delta 2 trans enoyl coenzyme A? Remember, this needs to be further oxidized, but this double bond prevents the oxidation, further oxidation of this trans enoyl coenzyme A. So first our body tries to get rid of this double bond. To get rid of this double bond, one water molecule is being added. What this water molecule do? This water molecule dissociate into H plus and OH minus. This H plus and OH minus, this OH minus binds with this delta 3 carbon and this H plus binds with this delta 2 carbon. So both gets new molecule and this double bond gets converted to single bond. So let's write down that resultant compound. So we have R, which is unchanged. With this delta 3 carbon, OH group gets attached. So RCH and bound, it's bound OH group. This next delta 2 carbon has one extra hydrogen. So now it's a CH2. And so as new molecules are bounding with both the carbon, this double bond gets converted to single bond. The remaining structure is unchanged. So C double bond O followed by sulfur and coenzyme A. So as you can see, now the carbon backbone has all the single bonds. And what we call when all the bonds are single? We call it as a acyl, and with this coenzyme A, so acyl coenzyme A. But this is not a typical fatty acid. This is the change, okay? So this OH group is attached to which carbon? It is attached to beta carbon. This is alpha and this is beta carbon. So it's a beta hydroxyacyl coenzyme A. So the name of compound is beta hydroxy acyl coenzyme A. And this reaction is catalyzed by the enzyme. Here once again, it is a name of substrate followed by its action. So substrate is enoyl coenzyme A, enoyl coenzyme A followed by the action. Action is the hydration. So enoyl coenzyme A hydratase. So this enzyme catalyzes and converts to the beta hydroxyacyl coenzyme A. Now next, what happens to this beta hydroxyacyl coenzyme A? As I told you earlier that these double bonds are problematic for the beta oxidation, right? So now that double bond is removed. So now this beta hydroxyacyl coenzyme A, it undergoes further one more round of oxidation. So this is the second oxidation. Now here what happens? Here also 2 hydrogens are lost, but both hydrogens are given by this beta carbon. One hydrogen from here and one hydrogen from here. So, and this hydrogen removal is facilitated by another coenzyme, that is NAD+. So NAD+ comes and it takes away 2 hydrogens. So NAD+ gets converted to NADH plus H+. And this NADH undergoes electron transport chain and it gives rise to ATP. Here also, as per new information, 2.5 ATPs are generated by 1 mole of NADH, and as per old information, 3 ATPs are generated when NADH passes through the electron transport chain. Now what will happen to this molecule after losing this both hydrogen? So let's write down its structure. So we have R, which is unchanged. Then with the carbon, it has lost its hydrogen, and this oxygen, it has lost its hydrogen. So now there will be a double bond between this carbon and oxygen, right? Then remaining structure is unchanged. So we write CH2, then C double bond O, and then S coenzyme A. So here also, as you can see, all double bonds are there, all single bonds are there in the backbone of this fatty acid. So we call it as a acyl coenzyme A. But here there is this keto group is there, and what is the location? This is the alpha carbon and this is the beta carbon, right? So there is on beta carbon there is a keto group. So we write beta keto acyl coenzyme A. So this is the name of this resultant product. This reaction from beta hydroxyacyl coenzyme A to beta ketoacyl coenzyme A is catalyzed by the enzyme, and here also the naming of enzyme is same, that first we have to write down the name of substrate followed by its action. So action is dehydrogenase. So the name of enzyme becomes first this beta hydroxyacyl coenzyme A. So beta hydroxyacyl coenzyme A followed by its action, that is dehydrogenase. Now, by the second oxidation step, what happens? This particular bond is become weak. It can be easily broken down. So, for breaking down, one more coenzyme A with its SH group comes into the action. It reacts with this compound, and as I told you earlier, that this coenzyme A has special attraction towards this carbon which is bound with the oxygen. So what happens? This coenzyme A binds with this particular carbon. So this bond will be broken down, and the resultant compound is R, then C double bond O, and with this carbon, this coenzyme A is bound with its sulfur. So S coenzyme A. Now after this bond is broken, so this particular part is converted to this thing, and this particular part gets separated. So separate molecule. Now here instead of CH2 we write CH3, then C double bond O, and then SCOA. If you remember, this is the structure of acetic acid with its coenzyme A, which is called as a acetyl coenzyme A. Why? Because this is the structure of acetic acid. Okay, so we write it as a acetyl coenzyme A. And what is this structure? Remember, this R is the hydrocarbon chain which is ending with this carboxyl group containing coenzyme A. So this is also a acyl coenzyme A. Now if you are wondering that we are starting this reaction with the acyl coenzyme A and we are ending with the acyl coenzyme A, then what is the difference between these two acyl coenzyme A? Remember, here the two carbons are lost from this acyl part. So this contains a -2 carbon. Now what happens to this acyl coenzyme A? This acyl coenzyme A undergo further beta oxidation to lose its 2 carbon, and this process will go on and on and on till all the carbons are lost. Okay, and each time beta oxidation occurs, it releases one molecule of acetyl coenzyme A and one molecule of acyl coenzyme A, which is 2 carbon less than the previous one. So ultimately in the last beta oxidation cycle, we get 2 fragments of acetyl coenzyme A. Now what happens to acetyl coenzyme A? Acetyl coenzyme A undergoes a Krebs cycle and it gets completely oxidized to carbon dioxide and water. But while doing so, while doing a complete oxidation by Krebs cycle, it also generates ATP, and as per new information, it generates 10 ATP, and as per old literature or old information, it generates 12 ATPs. Now this reaction is catalyzed by the enzyme thiolase. So these are all the steps which brings about complete oxidation of fatty acid. Now the question is that how fatty acid oxidation, beta oxidation, gets regulated. See, in this entire reactions of beta oxidation, this particular enzyme is the slowest acting enzyme. So that means we can say that this particular step is the rate limiting step. And generally it is observed that whatever step which is a rate limiting step, it is a target for the regulation. So this particular enzyme is inhibited by a malonyl coenzyme A. Now you might be thinking that what is malonyl coenzyme A? malonyl coenzyme A is actually intermediate of fatty acid synthesis. So basically what happens, whenever fatty acid synthesis is going on, at that time malonyl coenzyme A level increases and it inhibits this particular rate limiting step of beta oxidation. So by doing so, our cells make sure that whenever fatty acid synthesis is going on, beta oxidation should be stopped, so that it does not occur that on one side fatty acid synthesis is going on and on another side fatty acid destruction is going on. Because if both pathways are occurring simultaneously, it will be a futile thing for our cell. Now, one thing to note down over here, that this enzyme is not required for all the fatty acid. It is required only for the long chain fatty acid. It is not required for the short chain or medium chain fatty acid. So basically short chain and medium chain fatty acid, they are not regulated at this step. Okay. Now there is also a second checkpoint for the regulation of beta oxidation of fatty acid, and that checkpoint is over here, that is at the thiolase level. What happens? Its product, that is acetyl coenzyme A, it inhibits the thiolase, whereas its substrate, that is coenzyme A, it actually stimulates this thiolase. So basically acetyl coenzyme A and coenzyme A both works counterly, right? So it is said that acetyl coenzyme A, its ratio with the coenzyme A determines the function of thiolase. If this ratio is higher, then the activity of thiolase is going to be decreased. One more important aspect is that this acetyl coenzyme A, it actually goes and stimulate one enzyme of another carbohydrate metabolism pathway, and that enzyme is pyruvate carboxylase. This enzyme is of a regulatory enzyme of a pathway gluconeogenesis. This gluconeogenesis pathway synthesizes new glucose. So by this, if beta oxidation is going on, that means more of the acetyl coenzyme A will be present. So it sends up a signal to the gluconeogenesis that beta oxidation is going on, lots of ATPs are being formed. So glucose don't need to be utilized, rather use the machinery to synthesize new glucose. So that message is being passed from the lipid metabolism to the carbohydrate metabolism. Now let's see that how many ATPs are synthesized by complete oxidation of fatty acid. So the next topic is ATP counting. ATP counting. And for ATP counting, let's take up an example of saturated fatty acid, palmitate. This palmitate contains a 16 carbon chain in its structure. So let's draw this 16 carbon chain. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 and 16. We know that with each beta oxidation, 2 carbons are lost, right? So how many beta oxidation will be there? So first beta oxidation will break down this bond, second beta oxidation, third beta oxidation, fourth beta oxidation, fifth, sixth and seventh. So there will be total 7 beta oxidation cycle occurs. And by this 7 beta oxidation, how many acetyl coenzyme A will be released? So this is one acetyl coenzyme A, second, third, fourth, fifth, sixth, seventh and eighth. So along with 7 beta oxidation pathway, we get 8 acetyl coenzyme A from the one molecule of palmitate. We know that during beta oxidation, one FADH2 and one NADH plus H plus is getting released. So for each beta oxidation, we get one FADH2 and one NADH plus H plus molecules are there. We know that for each, as per a new information, FADH2 gives rise to 1.5 ATP, whereas NADH plus H plus gives rise to 2.5 ATP. And how many times beta oxidation is occurring? 7 times. So we multiply it by the 7, right? So 1.5 into 7, it gives to 10.5, whereas 2.5 into 7 gives rise to 17.5 ATPs. From the 8 acetyl coenzyme A, we know that one acetyl coenzyme A gives rise to 10 ATP as per the new information. So, as per new information, this acetyl coenzyme A gives rise to 10 ATP. But how many ATP acetyl coenzyme A are getting released? 8. So, 8 into 10, that makes up a 80. So, if we do summation 10.5 plus 17.5 plus 80, the sum of ATPs is going to be 108 ATP. But we should not forget that fatty acid activation, which is happening only single times for each fatty acid, and in this 2 ATP equivalents are used, 2 high energy bonds are used. Okay, so we write down a minus 2 ATP. Initially 2 ATPs were invested, so minus 2 ATP, and that makes total 106 ATP. So 106 ATPs are being synthesized from a single molecule of palmitate. This is so much energy. And if you consider old system, we will discuss old system also, because if you are reading old book or some new book still utilizing old information, this number might be different. So let's do that also. So here instead of 1.5, we have 2 into 7. Here instead of 2.5, we have 3 into 7. So 2 into 7 is 14 and 3 into 7 is 21. And here on the acetyl coenzyme A side, instead of 10, we have 12 ATP as per old information, right? So 8 acetyl coenzyme A and 12 ATP for each acetyl coenzyme A. So that makes 96. So 14 plus 21 plus 96. This total makes 131 ATP. And here also we have to deduct 2 ATP, which was utilized for the activation of fatty acid. So 131 minus 2 is 129 ATP. So these many ATPs, this number you may see in the old textbook, right? So don't confuse over there. Now I have one exercise for you. You need to count that how many ATPs are generated as per new information as well as for the old information. And the fatty acid you have to consider is the stearic acid. Stearic acid. Stearic acid is a saturated fatty acid and it contains 18 carbon. So write down your answer in the comment section below. And with this, I complete a beta oxidation of fatty acid. In the next video tutorial, we will see that what can be the defects or what can go wrong in the beta oxidation of fatty acid. I hope that all the concepts discussed over here are clear to you. If you still have any query or confusion, please write it down in the comment section below. Thank you.